I'm Chris Todd Hittinger. I'm an assistant professor of genetics and a researcher with the Wisconsin Energy Institute. Way back, I was a dinosaur nut as a kid, so I memorized all the names of all the dinosaurs, and I was really interested, especially in the historical angle of how these things came into being and how they changed over time. I'm an evolutionary geneticist by training. And I'm particularly interested in the genetic mechanisms by which organisms evolve. Midway through graduate school, I actually started transitioning into using yeast as a more quantitative system for addressing basic questions about evolutionary genetics. It turns out that if you want to study yeast as an evolutionary genetic model system, you don't have a whole lot of morphological differences to work with. And one of the major differences that you have is actually carbon metabolism. And these are traits that are really fundamental to the heterotrophic niche that yeasts inhabit, where they're not able to make their own food. Instead, they're breaking down food from the environment. In the case of Saccharomyces, they're only going to partially consume it and actually pump out a fuel that can be used by humans. I'm privileged every day to come into the lab and be able to look at how you assemble complex traits and how you can push a basic lifestyle plan such as yeast of breaking down simple sugars and other carbohydrates, and how you can create those variations on a theme that then lead to the differences in ecology and function that support hundreds and indeed thousands of novel species.